What we're going to get into now is the opposite of that. This is active transport. So now we're talking about movement of particles against their concentration gradient. And as you would imagine here, this is going to require some energy input, okay? So just to kind of really reiterate this here again, simple diffusion, facilitated diffusion, and channels do not require any energy input because they're moving according to their concentration gradient. In fact, they're going to liberate energy as they move according to their gradient. When we move according to active transport, we need energy. This needs an energy input in order to move that particle against its concentration gradient. So now we're moving from a state of low concentration to a state of high concentration, and that requires energy. It's not spontaneous. It's going to be that uphill type of movement that we described earlier, okay? Now, there are different types of active transport that we're going to get into, but in general, it's going to require a pump of some sort. It's going to require something, again, to help that particle move against its concentration gradient. And so in our example here, we've got the movement of particles. And so we're moving here from side A to side B. We've got a lower concentration. Actually, we've got equal concentrations here of particles on either side. But then down here, as we're continuing to move, this is going to require energy. We're now moving particles from side A to side B, which is against its concentration gradient. That is going to require some energy input, okay? Really important to make that distinction that active transport moves against the concentration gradient, so from side A to side B here, and that will require some type of energy input, such as a pump, and that pump will either use ATP or it will use the energy that is harnessed from another molecule moving down its concentration gradient. Okay, so let's talk about the characteristics of a pump. A pump is going to be, again, a transmembrane protein. It's another one of those proteins that are embedded into the membrane, and it is going to be able to harness energy from the dephosphorylation of ATP. So ATP is going to be the main driver. This is where we're going to get that energy from to move this particle against its gradient. So we're going to dephosphorylate ATP, and we're going to take that phosphate group, use that energy to drive this pump, and then move that particle against its concentration gradient. Now it's going to have specific binding sites. So it's going to have a site that can bind, one ion versus the other. A really good example is the sodium-potassium pump. And so the sodium-potassium pump has a specific binding site for sodium and then a specific binding site for potassium. And so it'll be able to move those molecules against their concentration gradient using the energy that's harnessed from ATP. Now, these will also exhibit saturation, right? These also have the ability to be maxed out. And so the rate of this type of transport will also be dependent upon the state of these pumps, whether they're maxed out, and then their individual rate as well. Okay, so now we're going to get into the two subdivisions of active transport. We've got primary active transport, which uses that ATP, and then we've got secondary active transport. And this is where, again, we can borrow the energy or, take the energy from another molecule that is moving along its concentration gradient. This is why it's so important to understand that when a particle moves down its concentration gradient, it is actually liberating some energy. And so we can kind of capitalize on that, use that energy and move something else against its concentration gradient. And that is what our secondary active transport is. So getting into primary active transport, the energy to move this particle comes from the hydrolysis of ATP. And so the main example, we're going to speak about this again and again in several different units, is the sodium-potassium pump, okay? And so the sodium-potassium pump can bind 3 sodiums, it can bind 2 potassiums, and it can basically move these particles against their concentration gradient. And I want you to think back to something that we talked about, the fact that there is more sodium outside the cell. Let's actually illustrate this here. Based upon our prior knowledge, we said that there's more sodium out here. So there's more sodium out here. there's less sodium in here. And so in order for me to move sodium out of the cell, that's going to be active transport. That's going to be moving against its concentration gradient. And this is where that sodium potassium pump comes in. Same thing for potassium. There's more potassium inside the cell, and there's less potassium outside the cell. So in an effort to move potassium into the cell, we're going to have to move it against its natural concentration gradient. And this is why we need that sodium potassium pump, because we need to be able to get energy to do that, to move both of these ions against their concentration gradient. Okay, so what happens here is we've got 3 potassium binding to the inner leaflet of this pump. ATP is hydrolyzed, and those are kind of spit out on the extracellular surface here. And then for potassium, the same thing happens. We've got 2 potassium binding to their specific site on the inner leaflet of this pump. Once again, we hydrolyze ATP to release that energy, and then we simply move those potassiums into the cell. Okay. Excuse me for 1 second, guys. My eyes are tearing up. I don't know, I'm not chopping onions, but I don't know why my eyes are turning. Okay. All right, so that is ATP. The main example here, the sodium potassium ATP pump. And the real important takeaway message is that we're moving against the concentration gradient. And for primary active transport, we require energy, and we get that energy from the hydrolysis of ATP. Now, as far as secondary active transport, again, we're still needing energy here, but the energy comes from a different source. The energy is released from an ion that is moving according to its gradient. So we kind of take the energy from another molecule. So again, in this example, we've got high amount of sodium here, low amount of sodium here. And so when sodium moves into the cell, it does so passively, hence this green arrow. So as sodium moves down its concentration gradient, we basically liberate this energy, right? And we take that energy, and then we can move something like glucose against its concentration gradient into the cell. Alrighty. Another good example of this, or another example of this, a specific example, is the sodium-linked glucose pump. And so speaking more specifically about this type of pump, it's going to harness the energy from sodium moving and then use that energy to move glucose into the cell, okay? And so again, this is moving glucose against its concentration gradient, but sodium down or according to its concentration gradient. And then this can give up the energy that this process requires. This is called co-transport, by the way, and this is specifically to sodium-linked glucose movement, because both of these particles happen to be moving into the cell. Another example of this is the sodium-linked proton pump. Now, this is called counter-transport. It's the same type of mechanism, so same thing is happening here. We're liberating the energy via the movement of sodium down its concentration gradient, and then we're using that energy to drive the movement of hydrogen ions or protons against their concentration gradient. The only difference between this and the previous example is that now the molecules are moving in different directions. So sodium is moving into the cell, the protons or hydrogen ions are moving out of the cell, and so this is going to be called counter-transport, whereas the sodium-linked glucose transporter is an example of co-transport, okay?
So is the co and counter just representing like the movement of it, like in the same direction or opposite directions?
Yep, exactly. So the co is going to be both ions, both particles or both molecules moving in the same direction, whereas counter is going to be they're moving in the opposite direction. So just the direction is going to be what determines co versus counter.
Okay, thank you.
Yep, sure. All right. Any other questions at this time? Any other questions? Okay. Here's a really nice summary table. And again, I'm going to encourage you to practice using this table as a study tool. So try to clear out the boxes and, just keep the column here and then the main row at the top, and try to fill in the details of this on your own. So think about the direction of net flux, right? The direction of net flux for simple diffusion, for passive transport via channels or via facilitated diffusion. All of this is going to be moving down the concentration gradient, okay? Whereas for active transport, whether it's primary or secondary, this is always going to be moving against the concentration gradient, right? Another column here, whether or not it requires energy. So we know that it doesn't require any energy for simple diffusion, channel, and facilitated diffusion, whereas for active transport, it will require energy. So practice just clearing out this table and trying to recreate this from your understanding of this, and then comparing this to this actual table. All right. Okay. Now the final slide here really shows that example that I kind of talked about earlier, which is really how everything fits together. We've got the sodium potassium pumps on one end. So this is that sodium potassium pump. It's pumping potassium into the cell against its concentration gradient. It's pumping sodium into the cell, out of the cell against its concentration gradient. So both of these are examples of active transport, right? Using ATP here. But then we also have different movement happening across the apical or the top surface of the cell. We've got sodium and potassium moving via ion channels. So this is an ion channel. This is an ion channel. And these are both examples of ion channel or passive transport. So we're moving potassium according to its concentration gradient, right? It's going to be moving from the inside of the cell where it's at a high concentration to the outside of the cell where it's at a low concentration. And so this is a passive process here. And same thing with sodium. Sodium is moving from the outside of the cell where there's more of it to the inside of the cell where there's less of it, and it's moving down its concentration gradient. And so what we can see here is that both of these mechanisms are working with each other. So essentially, this is called a pump-leak system. So potassium can leak out of the cell, down its concentration gradient, and then it can be pumped back into the cell against its concentration gradient. And so we have this leak, pump, leak, pump, leak, pump phenomenon, which is what keeps the relative concentration of potassium and sodium stable inside and outside of the cell, right? And if we were to think about it, if we only had one of these processes taking place, like if I was only leaking potassium and it didn't have a way to get back into the cell, then my extracellular levels of potassium would eventually start to rise. Same thing for sodium. If I was only pumping sodium out of the cell and it didn't have a way to get back into the cell, my extracellular levels of sodium will eventually rise. And so having the pump and the leak is a way to keep the relative extracellular and intracellular concentrations stable, even though we have movement of these ions in different directions, okay?